Hello!
Hi there.
Welcome to Microbiology Twitter Journal Club, where we care about all things great that are small.
My name is Danny, and in previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
And my name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but I also work as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today is our news week, where we do an overview of papers that we've seen in the news or other sources to try to find an article that we want to cover next week in more detail.
And next week will be our deep dive week, where we take one of the articles that we look at today and dive in deep, figure by figure, to learn what can and cannot tell us. So make sure to subscribe to satisfy your microbiology curiosities.
You can follow along with any of the papers we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet at us. This week we've got some. It's been a tough 2 weeks, especially. We're going to start off with a news story that's been happening in India. So, probably if you've been around YouTube, you probably heard a lot about the Indian coronavirus outbreak, and it's quite like harrowing at this point because I recall this time last year when all the healthcare systems around the world in the West were strained,
Mm-hmm.
and I'm thinking like how hard it was for us back then, and now taking that and saying how much worse it is in India and how much help they need.
Yeah,
So yeah,
absolutely. I
yeah,
mean, I think it's hopefully everybody has, or maybe hopefully everyone doesn't have that experience, but a lot of us do have the memory, right, of those early days of the pandemic when healthcare systems get overwhelmed. And it sounds like that's what's going on down there for a variety of different reasons. So yeah, I don't know how this is the huge challenge of like right living in the global community is like
mm,
how does one help anyways for these types of things? I'm not sure. I think there's definitely a sense of powerlessness that one can feel reading the news. But yeah, I don't know. We're gonna read some of the papers that have been coming out, I guess, from this particular outbreak.
yeah, and also the thing to emphasize this, we're very very early on the outbreak. So the paper we're talking about just got posted in the last week, and
Hmm.
it's still progressing. There really isn't much information out there that's been peer reviewed. So we're going to be going through a lot of preprints today, um, which haven't been peer reviewed, but they can still provide some interesting information. So the
Yeah,
first one
and I
will
think the
be
other thing is that like a lot like there's still all the signs that we've had in the past, right? Like the outbreak is not something totally new in the sense, right? Like that like there's some big shift in the way that the virus works necessarily, right? Like these are the same factors of epidemiology that we've right have experienced before. But there has been a lot of talk about these variants, right? Like as and this first one's gonna sort of talk about this.
yes, indeed. So I'll switch over to all right the paper reading view, where the first paper we're looking at is Convergent Evolution of SARS-CoV-2 Spike Mutations L452R, E484Q, and P681R in the second wave of COVID-19 in Maharashtra, India.
Mm.
Um, so yeah, this paper comes from on behalf of the of various government organizations working in India, and it talks about how eventually this kind of ballooned in Maharashtra, which is one of the second most populous state in India. Uh,
Hmm. Yeah, so
and
this means that they are like I think that this is something that's like kind of cool to know that because people know about the variants ahead of time, right? Like those surveillance programs can be deployed in these instances, and they can give us a snapshot of what might be going on, but not necessarily causality, right?
right, yeah. So this this comes from Indian SARS-CoV-2 Joint Genomics Consortium, and they did some sampling. A lot of international travelers get sampled, and they also sampled 5% of positive cases. So in the end, they collected about 733 nasal swabs uh, between November 25th, 2020 to March 31, 2021. So they get some idea of the timing of when this started.
Hmm.
Um, and so this preprint aims to catalog the different variants circulating in India around this time frame. And what they found was that the dominant strain was this strain called B617 that we're hearing a lot about in the news.
Hmm.
Let's see. they the main figure was in the supplementary material, but what they did was they also did like a lot of focusing in on actually the mutations that are found within this B617 lineage. And so
Hmm.
what I'm showing on the screen is like this tree map showing all the diversity of different mutations that have been kind of spotted across the different strains that have been sequenced.
Hmm.
Um,
And this is not like super totally new, right? Like we've seen these maps before, right, from the outbreak globally over time, and we also know when you zoom into a locality that has tons of cases, these types of mutations will appear.
yes. Uh, and the thing that this paper highlights is that there were two big mutations that people were quite concerned about: the E484Q and L452R mutations. So
Hmm.
these have been linked to antibody escape in previous papers. Um,
Yeah. I think I've heard about the E484Q. Is that one also present perhaps in the South African variant?
I think we've it's E484K. So this
Oh.
one amino acid gets changed to another. I think it gets changed to something slightly different, but I know that the E484 amino acid at that position. I can't remember what the E stands for at this point. But I know it's very
Lieutenant.
important. I will look it up later.
Yeah.
Um, and L452R. So the point is that these are both in the ribosome. Sorry, not the ribosome. Receptor binding domain. So that which we've seen is very important for the binding of antibodies to SARS-CoV-2 and also
Hmm.
binding of SARS-CoV-2 to its target ACE2. And
Hmm.
what they did here is they did some analysis to see like so they had the sequence so they can from that sequence they can build up what the protein looks like and test
Hmm.
out say how well it binds to ACE2 and also how well it binds to antibodies. So, I mean,
Hmm.
things like D614G was found. So that's the like Uber SARS-CoV-2 strain that's spread out. It was a mutation
Yeah.
that happened early on, and that was found to bind to ACE2 a bit more effectively, and that affects transmission. But also, they're concerned about whether it's more transmissible, and also whether it can avoid antibodies. So
Hmm.
what they did was they tested out on two like antibody treatments that we've. So they looked at the Regeneron antibody,
Hmm.
which was created by the company Regeneron. There's a lot of talk by Trump especially
Monoclonal
about that.
antibody. Yeah.
Yep. So they tested one of the Regeneron antibodies and modeled whether that could still bind, and they also
Hmm.
modeled whether another antibody isolated from a patient in Wuhan called P2B2F6 could bind as well. And what
Hmm.
they found was that actually they couldn't. So this led to the hypothesis that maybe this could be better at resisting the immune response.
Hmm. Yeah, I mean
Um,
this is similar stuff, right? That it's like another set of so right now we know from tracking variants around the world, right, that there are variants that are better or worse, right, to binding to monoclonals, right? I think that similar things like this are also seen in the P1 variant. They're not the same mutations, but they can occur, and to me, it just highlights this idea when you have outbreaks of virus, right? Like it drives evolution, and then those get selected for, right, for things that are better at doing one thing or another. Um, it's unclear, I guess, right, from the data they're presenting if these is like selected specifically because it's better at something, but that's sort of the assumption I think that's being pulled along with every variant that we speak about.
Yeah, this is based on research that's been published before. They don't actually test head to head with real antibodies. They model it in a computer to see whether they link up. And of course,
Mhm.
they only test two antibodies, whereas your immune response will evolve and develop all sorts of different antibodies.
Yeah.
So it's not necessarily conclusive proof that it will evade your immune response, but it is something that a possibility that we should be very, very aware of at this strain.
Hmm.
Um,
Hmm.
and
And
yeah,
I guess
and
like that's the that we should be aware for everywhere, right? Like if
yes,
if outbreaks occur again in an area, even if you have some sort of like, there's always the possibility of those mutations somehow, negatively impacting the immunity that the population has.
yeah. So I mean, this has now been banned. So I don't know if you've heard the phrase double mutant, but that's what a lot of people have been referring to this strain as,
Mm.
and it's quite a scary name. It implies like it's going to be doubly infectious, but actually, what it means is unknown at the moment. We don't know whether it's this. We've got little bits of
Yeah.
information, and we're inferring the worst case scenario because that's generally what we do in these situations. But really,
Mhm.
we don't know enough about this strain at the moment. Um, so that brings me to the next paper on this strain called uh, neutralization of variant under investigation B1617 with sera of BBV152 vaccinees.
So that's the
So
vaccine that the Indian government developed, right? Or it was like a company, right? They have one company,
yeah,
Bharat or something like this.
yeah, that's right, Bharat Biotech. So they're one of the authors of this paper. This is a vaccine that is commonly known as Covaxin. It's a heat killed. It's not heat killed. It's just a killed bacteria variant. So it's very similar to the Sinopharm and Sinovac versions of the vaccine.
Mm-hmm.
Um, so again, it's very much old school,
Yeah,
like how you make
classic way
virus
to
vaccines.
make vaccines, grow up
Yeah,
virus and inactivate it.
yeah, and I think there's been some preliminary evidence in the other versions and this one that it is quite successful at creating an immune response.
Mhm.
So it'd be quite interesting to see how it reacts. So what they did was they found, so it's currently in phase 2. So they took some serum from people in that phase 2 trial and checked to see like whether how it neutralized different strains of the
Yeah.
virus. So we've got this big. So I think I'm drawing your attention to figures C, D, and E because I think those are the ones that are most interesting. Um, so this is where they test against like the D614G variant, which is basically the variant that's taken over the world. It's the most common one at the moment.
Hmm.
Then it looks at the B617, that's the new Indian strain,
Yeah.
the B117, which is the UK variant, and then they test to see how well serum from people who have been vaccinated neutralizes them. And uh, it seems that they're so in C it looks like they're very comparable.
Yeah.
And D essentially shows the same data as C, except it's on a log scale, as far as I can tell. And and it's kind of and the lines are indicating that the serum comes from the same person.
Yeah.
So you can see like maybe in one or two people they have a low response to the new strain. Um, so it does seem to suggest that there is some immune escape happening there,
Hmm.
but not but it's not necessarily a lot that's happening. So
Yeah.
there is still an immune response going on.
Yeah, I mean, these are just neutralizing antibody, right? Like this is one of the things, like even with when the other variants appear, right? Like we've seen this type of data before. It actually doesn't
Mm.
seem as dramatic as some of the full decreases from some of the different serums. Like I guess I was recently looking at the the AstraZeneca's effect on the South African variant. That data seemed like it was quite much weaker. And so, like, there's
Mm.
a range, I guess, of vaccines versus variants in terms of how much reduction in neutralizing antibody titer there is. But again, that's only like a rough correlate of what that might mean for immunity, because it might not be that, like, just because that some antibodies are less good at binding to the variant, it doesn't mean that there won't be the right immune response being liberated. I mean, especially in light of the sort of articles we've been talking about with like memory B cells, right, and being very important in proper immunity, but in natural infection being somewhat modified. I feel like, yeah, it's hard to say. Actually, I feel much more confused about neutralizing antibody information now than ever before.
Yeah, I mean because it's only a small part of the immune system. Because so even if the antibodies don't necessarily work, there could be something happening going on with the T cells or
Yeah.
various other parts of the system.
And I think that
So
like in E, that's part of, like, right, like the wrench in the thought, right? Because in natural infection, they have just as much antibody as in vaccine.
yeah,
Yeah. So, but
so
obviously, I think the outcomes from natural infection are not good. So you don't want to, right? Like that's if you just evaluated on neutralizing antibody, right? You wouldn't be able to understand the value of a vaccine.
yeah, and at the moment, nothing I've seen so far talks about whether it's more transmissible or not. They haven't seen anything that tests that like to the degree that I'd like
Yeah.
to because it could be that this is a more lethal strain, but I think isn't the other possibility that make it more dangerous would be if it's just much more transmissible.
Yeah.
Um,
And again, I think that in some ways these are, right. We're trying to find the question also could be why these papers about variants, right, and the outbreak, right? Is it trying to assign causality? Is it trying? It's trying to get ahead of something. I think that that's usually the case, right? It's just trying to say, could there be some complications, right, in a plan, right, by these variants, and that's kind of it's very interesting. I think from the when you think about like how do you deal with the outbreak of something, like given that we now know that when an outbreak occurs a variant is likely to come along with it just because of the amount of infection that's going on. So these types of preliminary pieces of data, I guess, are very useful for policy making and stuff to say, okay, well, maybe we don't want to use this vaccine, but they don't really test any other vaccines in this paper. So it's still just like a you have to take this data probably with other pieces of data, right, and use it together to try to inform a decision.
Yeah, and another thing I want to like kind of it's interesting for me and I guess for people who are working in this field that this most of these coronaviruses are being classified along the pangolin classification. That's why you get the B11 kind of designations.
Mm,
But the thing in B1167 I want to point out is that there is a lot of genetic diversity there that we're
mm,
seeing. And so I looked into this, and in the study they say that they took 146 COVID-19 sequences, and of those only 15 had the L40 had the double mutation.
hmm.
So
Oh, so
that
even
that's
this particular variant that people are focusing on is not necessarily the most prevalent variant.
well, I mean, again, this is between January and March, so with the April data, it might show it taking over. But
Mm-hmm.
I found that quite interesting, and it's so we might hear about under a new name soon, perhaps.
Mm.
Um,
Yeah.
but yeah, and I'm going to throw like another wrench in the works, I think, because we've got another paper. So we're talking about a lot about variants and their ability to perhaps break through. So
Mm-hmm.
now we're going to look at a study that investigates that. So vaccine breakthrough infection of the SARS-CoV-2 variants.
Mm-hmm.
Um, so yeah, it's a big worry about whether these variants can get around vaccines. So this paper from Rockefeller University in New York aims to study this in question. So they looked at 417 people who worked at university who had been vaccinated with the mRNA vaccine and who had the vaccine in 2 weeks previous. And they found that of those people, two patients managed to show symptoms of SARS-CoV-2.
Mm-hmm.
And so it's basically like it's like a case study essentially.
Yes.
And so they got like a healthy 51-year-old woman, 90 days after second dose vaccination, got some symptoms, and then those symptoms gradually declined. Another person was 65 years old who
Mm-hmm.
caught it off their unvaccinated partner and also had some symptoms, but then again, they eventually none of them had severe disease.
Yeah.
Um, and this one had E484K mutation as opposed to the Q we've seen before,
Mm.
and the other one had an S477N. So they both had different mutations, and they were distinct from the known variants that people were concerned about at the
Mm-hmm.
time. And the thing that really throws a wrench into the work is that both patients had high levels of neutralizing antibodies,
Yeah.
and they even tested the levels of neutralization and found that actually the patient's antibodies did neutralize the strain that they were infected with. And these serum samples were taken about 4 days after the onset of infection. So these are vaccine elicited antibodies that seem to attack the SARS-CoV-2 just as well.
Yeah.
So
But the
that
people
means that
still develop some mild symptoms, right? Like not enough to put them into the hospital.
yeah. So this is an interesting one because it means it's because I mean you can look at this paper two ways. You can say that these variants found some clever way to get around the immune system, or that just because of the way vaccines work, sometimes immunity takes different levels of time to come up,
Mm-hmm.
and also the other way you can look at it is that these people could have had much more severe infections that were dampened by the vaccine.
Yeah.
Uh,
I think
so
that would be one of my interpretations, I think, from it. I mean, this like we know that like the vaccines were never purported to be 100% effective, right? That's why
mm,
like right the models of controlling outbreaks shouldn't just rely on being vaccinated as as the thing that keeps everybody safe, right? There's still the pandemic is still going on, and even with everybody vaccinated, like there should be some care taken because we don't 100% know the dynamics, right, of infection, especially considering variants and stuff. But we have some pretty good indications that the vaccine is going to be a very powerful tool, right, in allowing us to return to some different form of living. So by studying the breakthrough infections, maybe we can learn more, right? Like I think that that's maybe the ethos
Well,
behind this paper is that like let's look at these exceptions and we can maybe come to some sort of thought about what we're trying to avoid, right? Like how do we limit even these things, or is this even a problem? Would this be a problem? Would these two people, right, stand like drifting around in society, right, having this infection, perhaps transmitting this virus, right, that was selected in them because it was able to go through vaccines, will impact our population, will impact like like sort of more community level immunity. I think that that's maybe where, of course, this paper doesn't answer those questions, right? But it's the sort of piece of data that might be helpful in advancing that type of thought.
yeah, it's it's an interesting thing to look into just because we want to know how the immune system works is to
Mhm.
create better vaccines, and finding out where something doesn't work, or that's a way to build on and develop for the future.
Yeah. I mean, I
Um,
wasn't even thinking from the developing vaccine standpoint. To me, I'm thinking from the uh, like sort of selection of selection of viral variants perspective, right? Like
yes.
these variants can exist potentially. I'm not sure, right? They don't show that at all, right? But the possibility exists because these two people were able to carry these variants, neutralizing. It still neutralizes, right? But presumably they're still coughing up this virus, right? But again, that's not directly tested here. But like this is the place where that line of thought would grow from.
Right. So it'd be something to look out for in because the thing
I
about
think it
variants is you
helps with
want to see.
surveillance, right? Like it's a certain
Yes.
like oh, here are two, like now that we know these two things, let's make sure that when we're doing our surveillance, because can't say it enough. For science, you only look at the things you can only answer the questions you've actually asked, right? You can't answer like very open-ended questions with science. You can try to get to those answers, but you don't like 100% get there from the data. And so to me, it's like surveillance measures are looking across the whole genome. Are they doing full genome sequencing? Are they assigning significance to the same loci the same way, right? In terms of when do you throw up the flag and say, here's a variant that's growing and we need to address it, right, with our public health partners. And I think that studies like this can help inform that type of surveillance work, right? Because if we are doing genomic surveillance, we are looking for variants of concern. Here are two mutations that might be important there, right? Still pending a lot of other follow-up information.
Yeah. Um, so with that, we're going to go on to another. So I'm going to throw another strange paper at you, but this paper is called "Time-Dependent Heterogeneity Leads to Transient Suppression of COVID-19 Epidemic, but
Mm-hmm.
Not Herd Immunity." So this paper is interesting because it asks people to rethink the idea of herd immunity and how it happens.
Mm-hmm.
Because I think there's been a lot of like almost premature declaration of herd immunity for we
Yeah.
reach a peak and then you feel like, oh, we're fine, and this is over now, and then a second wave comes up that's even bigger,
Mm-hmm.
and so I think quite a lot. I mean, I've been in the country that's been hit with that a number of times. So,
Mm-hmm.
uh, and this paper kind of like tries to answer how that could happen, even in countries where there haven't been many lockdowns, perhaps.
Yeah.
So, yeah.
No, I think it's I think to me it's a good warning type paper. Like the interpretation of this paper against a misconception that people might have, right? Where they say, I've seen the rates go down. I know the vaccine rates are going up. They must be they must be causal, right? Like, but actually it's correlation, right? Because we don't really like we don't really know that that's the factor that's driving the rates down, and so then behavior might change, then the rates will go up again. Will we say that the vaccine isn't working because of a variant? Or is it just because we never reached the herd immunity in which that behavior was supposed to change to the level, right? That would I guess prevent some level of infection. And so we're seeing that those behavior changes are impacting. And so like what I like about this paper is that it's a model, so it doesn't even know the mechanism, right, in
Mm,
which that these things can come up. It's like very it's very mathematical, right? Something, right? Like something could drive these different waves. And until you reach actual herd immunity, right, where you know that certain number of people are vaccinated, the ups and downs of infection could just be based on these some physical element, right? Heterogeneity, it sounds like, is what they're talking about, right? The fact that
Yes.
you get different locations with different outbreaks. That I haven't skimmed that much.
Yeah, I skimmed it, and they basically classify heterogeneity as the differences in people. So not everyone is the same. People have like different susceptibilities to the coronavirus infection, and also different social contact. Because coronavirus doesn't just like spread like out in the air; it's between people. And so
Mm,
if you are an active person who meets a lot of people, then you're going to end up like causing a super spreader event.
mm,
But if you're kind of very not like that, then you're less at risk. And depending on how your social network is, and say how many of these super spreaders you know, it changes. And so you can get this period of
yeah.
transient collective immunity where you get enough people in a certain community who have like shaken off or are not susceptible to the virus, that you can get this point where it seems like there aren't any infections going on. And
Hmm.
on the
Hmm.
other hand, like you can say that, okay, once we understand how this transient collective immunity works, we can actually use that to modulate how we shut down and lock down to try and
Hmm.
uh
Sure. Yeah,
so
I mean,
so
this is good data that informs like localized, right, lockdowns, and like if outbreaks will be cropping up, maybe this sort of information says, oh, let's tune those parameters then, right? Like if everyone can be classed by, I'm seeing figure two now, right? Like they have like this like threshold of heterogeneity, right? That like
yes.
in different, if you assume these different lambdas, I guess, is what's doing heterogeneity. If you assume these different lambdas for people, you get different outcomes in terms of across a range of R naughts. Um, and so
Yes.
yeah, like it gives you this idea that like, well, we don't know this, we know that these types of things exist. How do we change those lambdas maybe, right?
Hmm.
In specific areas because they're applied only to specific populations. Yeah.
Yeah, I mean they try to fit their model to New York and Chicago and try to indicate whether once lockdowns are lifted, what are the chances of another wave coming. So they're quite optimistic about New York. They say that, okay, New York had it very, very bad early on, so if the lockdowns are lifted, it might have it better. But Chicago, there's still quite a population that could be affected by that.
So
So
they're very much modeling behavior then more than
yeah,
anything, right? Like there's like if the assumption of like if the data that went into it is basically predicated on severity of the infection before, right? It's very much like a very high level view, right, of what human behavior may be like, uh, predicted by the severity of the infection you've seen in your local environment.
yeah. And it's interesting because it does focus a lot on the social dynamics of infection, a lot more than we've seen in. I mean, we did review a paper which took mobile phone data and collated it to infection outbreaks, but I think even
That's
this
right.
it takes that a little bit further to create this more synthesized model. Um,
Well, actually, I think that's fat. That now that you brought that up, that actually just makes me think that'd be really cool, right? Like maybe they could validate pieces of their model, right, by incorporating that mobility data as some sort of metric, right, of as
yeah,
like a metric for what they're doing. Yeah.
yeah.
Anyways.
A lot of this paper is very much above my skill level to understand at this point, so Uh, I, I see words. I don't necessarily understand all of them. I got the gist of it, but I think it's a very very interesting paper that I would like to
Yeah,
under
absolutely.
because
And again, I appreciate it's always good, I think, to remind everyone, right? Like that the world is a complicated place, right? And that just because like one technological achievement, right, seems to be doing well, it doesn't mean that there aren't all these underlying factors that could easily tip, right, the outcome of the future.
yeah. So I guess it's always be prepared for things that might happen in the future. Um,
Mm-hmm.
okay, so next paper is single cell multi-omics analysis of immune response in COVID-19. So if you've been following us for a while, you know that I have a particular obsession with single cell transcriptomics.
Yeah, because it was
Um,
technology that was, I think, more in its infancy when we were going through grad school, and now to see like this amazing amount of data that's coming out of these sequencing machines, and even able to sort of like get assorted by like they're doing cell type, right? They're able to get to this resolution where organs are made of multiple different types of cells that all have these distinct profiles.
Yeah, and this paper gets to a question I've been asking, like, I guess, throughout the outbreak. What does a good immune response to SARS-CoV-2 look like?
Hmm.
And this takes blood samples from patients from around the UK who've had either mild, moderate, severe, or critical SARS-CoV-2 infections. And it takes blood samples to look at what kind of immune cells are circulating around them, what kind of genes those cells are kind of expressing, and how they're interacting with each other. So this is kind of just like
Yeah,
what
and
I've
it's like
been
a
very interested in
higher,
this.
it's like by using this technique you get many more layers of detail. So the signal for what's good or what's bad is like right? Like you have more opportunities to find it, right? In the mix of all the comparisons. I think that that's maybe a good way to explain why using single cell multi-omics would be good against this one question, because we've seen this type before, right? And you can build associations, right, to what makes a good outcome, but it's like this is a very detailed view of what makes a good
Mm.
outcome.
Yeah, and it is very much like it depends on which patients you're looking at because people have variation in their immune system.
Hmm.
But the power of just taking every single immune cell and just asking it, "What are you doing?" is something that's very seductive to me. I'm very interested in that.
Yeah.
Um, and I fully expect that someone will do a takedown saying that okay, we're all making a bit too much of this, but at the moment I'm hype about this. So, yeah, it'll be an interesting paper to explore, but also we've got some interesting treatment papers. So, ColdZyme maintains integrity in SARS-CoV-2 infected airway epithelia.
Yeah, this one's kind of an interesting paper that I saw. I guess this was already in the works, right? It's not particularly made for SARS-CoV-2, but they have this product called ColdZyme, which
Mm.
um, essentially puts enzymes into your airway. Uh, Trypsin, I think, is what they do.
Yeah.
Um, and it was shown that it helps increase like the integrity of your epithelial cells in that area. And in this paper, they're investigating that effect specifically with SARS-CoV-2.
Yeah, so ColdZyme appears to be a generic kind of. It was designed to cure the common cold. So you take
Yeah.
it as a mouth spray, inhale it, and it forms a physical barrier that interferes with the entry of common cold viruses,
Mhm.
which get trapped in it. Um, so they also use some kind of primary cultures where they grow kind of lung cells in a petri dish.
Yeah.
Um,
Is really beautiful images.
yeah, yeah, I'm just looking through them. They they're very nice images. I have to say they definitely look good. But yeah, it's an interesting treatment. I mean, I'm kind of skeptical about these sorts of things because you really have to. Its timing is so important for this. Is it something you take every day, or is it something that someone coughs and you take off? Except you can't take it afterwards because that's so you have to like it's a
And
ongoing preventative.
it really doesn't really say anything about the outcomes to disease. That's the
No,
other thing, right? Like
yeah,
it's like they zoomed in, right? Like to the mechanism of action of this particular thing. So I mean, this is kind of the same thing as when you judge a vaccine based on its neutralizing antibody titers, right? It's like, yeah, that's all fine and great, but like, what does it actually say about disease in this case? Because when you have to deploy something against a disease, like that's a complex process. Like maybe you block these interactions, but some still happen, so you still get disease. How much is that contributing to that overall phenotype? It doesn't get at that.
yeah. So yeah, it is still very much like we've got something in a petri dish. We add something to it. But it's a nice thing in petri dish. It's a very interesting thing in petri dish. But um, I'm not sure
Yeah.
whether this is necessarily going to solve the SARS-CoV-2 outbreak.
No,
Um,
no. I mean, I think that, there's also this product already exists, right? So this is
right.
also the and maybe we haven't covered so many of these different things. I mean, no, it happened with remdesivir, right?
Yeah.
Remdesivir already existed, right? And they're like, oh, can it be used? And then you do a bunch of these trials, you're like, sort of, it can kind of be used. It's not like the best tool. It's also really expensive, right?
Yeah.
And like that's kind of what happens. And so here's another example, right, of like a really cool technology has the potential to do something. Do we really know whether it's going to have that impact? We have to hear more. And then many other factors will impact, right, its utility actually within our society.
Yeah, I'm sort of skeptical about this, but but I mean, they do some cool technology with the differentiated airway cells.
Yeah.
But yeah, next one. No survey, thanks. Saliva two-step rapid detection of asymptomatic SARS-CoV-2 carriers.
Yeah,
So
I love this paper. It seems very exciting to me. So like I knew that lots of people were working on this LAMP technique to
mm,
detect SARS-CoV-2 because we've probably previously talked about like RT-PCR, right, is the big way, right? You get nose swab, and they put in some machine, and then it amplifies stuff, and then you get a curve, and then based on the curve, you see how much virus you might have. Well, that's kind of difficult to do, right, without a lab. And this type of the type of assay they develop here, well, first of all, it doesn't need nose swabs because there's been all this information saying that saliva is just as good at detecting viral load inside of people.
hmm
And secondly, it doesn't use RT-PCR. It uses something called LAMP. And LAMP is a technique that it is basically a PCR technique, a type of PCR, an amplification technique, but it only happens at a single temperature. So you
right.
don't need a heat block. You can just incubate things at one temperature, and then it kind of comes up. And so there's some technological limitations to using this particular, and also the readout they use is color, right? So you don't even need a
Mm,
special reader to read this test; it's just color. But they sort of there are some technical limitations to using saliva in these color tests. So
hmm
this is just to me like a nice compendium that kind of catches everybody up. If you've heard about LAMP technologies out there, this catches up. Here's how it can be used, right? To test people. And I love the focus on asymptomatic SARS-CoV-2 carriers because I think that speaks a lot to the current world that we're moving towards, right, with increased
Right.
vaccination, is that we still need tools to manage, right, and understand, right, when will an outbreak occur, if an outbreak even can occur, right? And this is sort of, to me, an important technology to that end.
Yeah, I like it. It's a very practical technology because they are thinking about how it's going to be used in the clinic. It's not trying to be too fancy,
Yep.
and you can just see that by the fact that their graphs aren't even graphs; they're photographs of their experiments, which
Yes,
is
yes. Which
which
is in
I
some ways it train like if you're somebody who's interested in deploying this tech, like you get this like inbuilt training, right? Where it's like, like this is what I'll see if I do it, right? Like this is what I
yeah
expect to see, right? Like and you can see too like how there's like little variants too in the duplicates that they do, right? And like that can also I love it as a training guide in that way because you can look at it and be like feel more confident that like, yeah, they saw this too, right? Like this is how they made their judgment calls of the data.
yeah. I tend to like seeing numbers and things like statistical significance, but actually when you've got some things like color change and the differences are this stark, this is actually a really good way of demonstrating a way that's actually kind of a way that most people actually interact with this technology.
Yeah, yeah, and I mean you can still quantify it, right? Like you
Yeah,
can
you
still
can.
like in the dark days, not the dark days, in the past of microbiology, people also quantified turbidity by eye,
Yeah,
right? You just had like a bunch of standards that were kind of different levels of cloudy, and you're like, this thing is kind of cloudy between this cloudiness and this cloudiness. So like
true.
the human eye is good for certain types of outputs.
Yeah, in this case, you don't necessarily need to see like, oh, is this not yellow and is this pink? And is that statistically significant? Is that statistically significantly different from pink? I mean,
Yeah.
it's so yeah, I can see why they showed things like that. But yeah, I mean things like what was it I can't, the area under the curve, ROC curve would be
Yeah.
a nice sort of thing to have, but um
Yeah,
yeah.
do they do that? I guess yeah, they don't ever build that in this paper. Maybe it's in a supplemental. I'm not sure.
Yeah, it might be, but yes. So yeah, it. I mean, I like it because that hasn't got maths, but I remember that maths is necessary, and like my vegetables, I need to eat it with every paper. So um,
Yeah. And I guess the other big part of the work that they do here is that they also compare it against an RT-PCR test, right? So
right,
like there is a gold standard test out there. In the final figures of this paper, they do that comparison, and it seems just as good, I think.
yeah. Uh, so yeah, yeah, it does look like it does pretty well. Oh,
Yep.
some of the figures are really well made. But
But we've talked
yeah,
about nice figures in eLife before.
yeah, eLife just knows what it's doing when it shows papers. The papers are so easy to read on eLife. I really appreciate that. Oh, no, we've lost our signal. A minute.
Oh, oh, we've lost our slides.
Yeah, sorry. 1 minute. Wait, back. hold up a second. I'll need to reestablish them. Let me. Uh,
Okay.
okay, hold on a minute. Okay. So this is gonna take a while. I'll
Should
one
we
minute.
continue? Like, I can talk about the next one, or
Yeah,
no?
you
Let's wait.
can. Yeah, okay, that'd be great if you could while I get the slides back. Uh, okay, so what's the next one's name?
The next one we have is safety and efficacy of single-dose 8026 CoV2S vaccine against COVID-19. So this is the Johnson and Johnson, I believe.
Yeah,
People have heard about. I'm sure people have heard about the Johnson and Johnson vaccine out there. It's based on a very similar technology to the AstraZeneca, the Chadox vaccine, in that it's an adenoviral vector that delivers the spike protein coding sequence right into some of your cells. Your cells make it, and then thus immunity happens. I guess what's kind of cool about the Johnson & Johnson one is when they did their clinical trial, they only gave a single dose. And so that's kind of convenient. I think that that's probably why they did it, is because there's this convenience factor to it. Plus, if people remember from the Chadox vaccine, um, uh, adenoviral vaccines are very stable, so they can be stored at like 4 degrees, something like that, and so you eliminate some of those cold chain requirements. Um, and so that's another great thing about this vaccine. And it's already in the U.S. I'm not sure what it's like over there across the pond. Um,
it's not here yet. I mean, I think it's I think it's been approved, but it's just the timing of it. So yeah, I finally got the slides back. Hopefully they're going to be displaying. Oh, one minute, need
I
to.
see them.
Okay, yeah. Okay. Yeah. So yeah, as you said, I think it's one dose. Just looking through the data, it seems to be. Let's see. We got like some of the numbers here. 86 out of. Sorry. So the interesting thing is they also like look at the test on the South African variant, and they find that it's slightly less efficacious, but they still find some efficacy in it. What other things are there? They also looked for venous thromboembolic events. Sorry, thromboembolic events. So that's a common side effect that we've been seeing in lots of SARS-CoV-2 vaccine candidates.
Yeah.
Um,
I mean, especially these adenovirus ones, right? Like, I think
yeah,
that that's the big important thing, is that because it's the same platform of something that has already had some safety scrutiny, then we want the same safety scrutiny applied to this one as well. Even
yeah,
though, right, as we've spoke about in previous streams, that safety risk is very, very small, and it's something that occurs as you scale up. But again, like making sure that we know those risks ahead of time, and that doctors have that communication so they know what to look for when they're giving the vaccine, and when it's being deployed. And so when people take it, they also know the risks that they should be looking for in order to go to the hospital, if required.
yeah. Going for, and we're going. So that's a big thing, but there's even I think even bigger vaccine news if I can find it. Because I think yeah we.
The
So
next.
the next one is a paper called High Efficacy of a Low Dose Candidate Malaria Vaccine R21 in one adjuvant matrix M trademark with seasonal administration to children in Burkina Faso. Um, so yeah, we haven't talked much about malaria because malaria is it's it's a big disease that affects lots and lots of people, and this is a double-blind randomized control trial of a low dose protein based on R21, which is like so
A
this is
circuit.
does a lot circumsporozoite. Um,
So
so
like I think this is gonna get crazy because we haven't really even talked about like eukaryotic pathogens, right? Like
no,
you can imagine that as organisms get bigger and start doing a lot more complex behaviors, you get like really complicated life cycles going on. So malaria is a eukaryotic parasite, and so it has like a whole life cycle, right? Where it has like
yeah,
egg form and like sexual form, and it like swims in your bloodstream, and then it goes into red blood cells. It's being classically thought of as a very difficult organism to vaccinate against because um, because of all these life cycle changes, like of course the surface chemistry of this organism is changing constantly. Furthermore, it like is in your bloodstream, and I think there's variance. It can change up the coat that it has. So it's been very difficult to vaccinate against. And so this is big vaccine news because this is like one of the first good news vaccine efforts that have come out, I think, for a malarial vaccine.
Yeah, and so this focuses on children because children tend to be like the target population for this. I
Mhm.
think the vaccines tend to be a bit more efficacious in children, and children under 5 years tend to be the most vulnerable groups affected with malaria. And in some high transmission areas, partial immunity to disease has been observed through childhood. So
Yeah.
in this study, they find that they get about 70 between 74% on average, so 63 and 82 for which is
Hmm.
amazing efficacy
Yeah.
compared to previous vaccines, which seem to be a bit lower.
Mhm.
So, I mean, the current malaria vaccine has about 33.6% efficacy in adults and 55 efficacy in children. So, this is a big jump up.
Yeah.
Um,
Yeah, no, it's
so,
really cool. And also, this disease is like primarily in warm weather areas, but with climate change, it could also be coming to your neighborhood near you.
yeah, I'm talking to you, America, and other places that might become warm. Um, uh,
um, so
but yeah,
yeah, but I think it's actually very exciting to hear that this is going on, and I think like we've been thinking a lot about, or the public attention has really been on vaccines because we've been waiting for this SARS-CoV-2 vaccine, the SARS-CoV-2 vaccines. It's also interesting to just take some of that knowledge that we know from distribution systems and say, does this vaccine stack up, right? Like, it, as we as people read this, as people hear this news, I think it's good to also ask like, how is it going to get distributed, right? Like, does it have cold, right? Does it need cold chain? Like all those things would be important because it would be great to get rid of malaria. I hear that it is not a very good disease, and it falls in that neglected tropical disease category because it doesn't happen in like wealthier nations as
oh
much.
yeah. I mean, the anti-malarial drugs are bad enough, so the malaria itself is really not a good disease to
Hmm.
have to deal with. Um, so coming off the vaccine news, so we got a comment about I think 2 weeks ago about someone who's concerned about vaccine side effects and Bell's palsy.
Mm.
So I decided to go on a little like kind of dig into the story about Bell's palsy, and so I'm gonna just present that. So
Yeah.
firstly, Bell's palsy is an annoying disease, but it's not fatal.
Hmm.
It's temporary weakness in one side of the face, which usually resolves in a matter of months, but leaves you looking a bit odd, and it can lead to some pain and some other things.
Hmm.
So it's not. So I hadn't heard about. So this commenter pointed it out. I hadn't heard about the story, so I looked into it. So you don't. So it hasn't been reported too widely. So, so I'm starting with looking at the emergency use authorizations. They
Mm.
detail some cases of Bell's palsy. So in I think the Moderna they see I think 4 cases of Bell's palsy. Oh no,
Hmm.
sorry. In the Moderna they saw 3 cases of Bell's palsy, and
Hmm.
in the BioNTech they found 4 cases. And here's the thing that because I'm going to talk a little bit about adverse events in general because
Yeah.
people have complicated lives and a lot of
Yes.
stuff can happen irrespective of. So I mean, I had cereal this morning, and then I might get a cold. Doesn't mean the cereal gave me the cold, and
Uh-huh.
that's always the kind of trouble is trying to pick out the causality when someone's getting a vaccine because people have lives and people do other. So for instance, so they looked into this Bell's palsy story. So we don't really get much detail from the Pfizer one, but Moderna goes into a lot of detail documenting the cases. So, for
Hmm.
instance, one case it was a 67-year-old female with diabetes who was also like hospitalized for stroke. Another person had some people had unrelated respiratory infections. Other people had other underlying diseases. So from these initial like kind of findings, it's very difficult to tell whether coronavirus, the vaccine, was causing it, or these other underlying issues.
Mm-hmm.
Um, and I think yeah. So I mean, I think one thing that we noticed in the AstraZeneca paper was
Yeah.
people who like got the vaccine were less likely to be in road traffic accidents or shot with guns.
Mm-hmm.
Uh, but no one's going to suggest that the vaccine protects you against being killed in a road traffic accident or being hit with a gunshot. But
Yeah.
so we have to almost take that same energy when we look at these kind of side effects because there's going to be a lot of fear mongering out there.
Mm-hmm.
Um, so I'm going to talk about this next paper, which follows up on the story about facial paralysis with,
Mm-hmm.
and what they found was that so this is a paper that delved into that question in a lot more detail, and they basically compared the amount of facial paralysis like cases that have been linked with coronavirus vaccines with other vaccines, and they found that they're actually about the same, and in totality they're about the same as the overall like population. So
Ah.
if it was linked, we'd actually expect to see it be a much bigger spike in facial paralysis cases. Because if we were to extrapolate from the study we saw, uh, from like these end use authorizations that I just
Yeah.
showed, we'd actually expect it to be much more prevalent than, say, the coagulation side effect that we have been seeing. That is a
Gotcha.
real side effect. Um,
Ah.
so, so
So
at
this
this
one is
point,
just the initial data had some indication that this could be, but they didn't look into the larger population and really dive deep, right, into the comparison. And so when they did that, it doesn't seem like it is associated with vaccines at all.
yeah. So, I mean, we're going to see a lot of things like this, where a lot of cases where people come up with have experience a disease after like getting a vaccine, and then it's easy to make that causality link.
Yeah.
And there's going to be a lot of scaremongery that'll happen due to these kind of coincidences, where people get the disease experience of being the equivalent of being hit by a car after walking out of the vaccination center, except it's a disease. So we can't as
Yeah.
clearly say that it's not related to the vaccine. So there's always going to be more investigations to chase up each individual thing.
Hmm.
And and so I mean I've been seeing a lot of papers that seem to be in the region of scaremongering about these vaccines. And the
Mm.
counterexample of this has been the way that people have been writing about the clotting disorder, which is a genuine side effect. And we've reviewed a couple of papers that have come through. And in
Yeah.
those cases, they didn't just say, "Oh, there's a side effect." They also came up with how we're going to deal with it, what symptoms to look out for, what treatments to do if that happens. So there's a
Yes.
difference between scaremongering and trying to understand the side effects, and
Yeah.
it's going to be quite difficult because we're going
When,
because
when you say that you've seen papers about this Bell's palsy, or do you mean articles? Like
oh,
just news
I
articles,
saw like two case reports. One
case reports?
was one person who got Bell's palsy after being vaccinated.
Mm.
I decided not to share that because the the authors misrepresented what was written in the emergency use authorizations to make it sound more severe than it was.
Gotcha.
So they said because in the EUA they spotted cases in like the vaccinated and unvaccinated group, and in those papers they said, oh, these cases, and they lumped them all together as and said, oh, the vaccine's
I see.
responsible for all these, which is actually a real uh,
Hmm.
sneaky move to try and make things look more scary than they are.
Yeah.
So again, that's fear, that paper falls into the aspect of fear mongering, where
Hmm.
it's not about trying to get you to understand disease; it's about to get you to be afraid and feel like that person who's telling you that information is more important. And it's
Yeah.
very and I everybody kind of has experienced that to some extent during this outbreak. So
Yeah.
um, and then we have the Greinacher paper, which papers now that are studying this like the side effect, and and they find a lot more cases, but we see a lot more care of the way they talk about it,
Ah.
and kind of advise how to deal with these cases.
Sorry, I
Um,
don't know what paper you're referring to when you said
oh,
the.
so we looked at preprint by Andreas Greinacher, so the one where they looked at the clotting disorder and found the
Gotcha.
TF4 antibodies,
Yeah.
and then suggested treatment.
I remember now.
Yeah.
Yeah, no, I mean, I think that like, this will come up. I heard it said, right, like because the news cycle for SARS-CoV-2 is so fast and furious, right? Every small scientific thing can like blow up and become like this could be the next, like watch out for this thing. But then, like it takes some time for science to accumulate and for more for people to publish their studies to really pin down what's going on. And yeah, it sounds like from the story you're telling me and this paper here, I mean, I can see it for myself, right? That yeah, if you're comparing the rates of Bell's palsy, like inside and outside the vaccine groups, there really isn't cause for concern, or there isn't a big cause to link the causality. There isn't a big rationale to link the causality, right, to the vaccine.
Yeah, and with science, we're always talking about like whether, because there's always some uncertainty with science, but at this point we're saying that the signal, if there is a link, it is so rare that it's almost undetectable.
Yeah.
So it might be that there might be one person who is the unlucky person who gets this. But then again, Bell's palsy, it's not a nice disease, but at least it's not fatal. It's not long lasting. You do recover from it.
Yeah.
So
I mean, I don't think there's side effects for all sorts of drugs that are pretty terrible, right? Like, so in some ways, we've talked about this before, right? When we just discussed adverse events, right? Like, there's a risk benefit analysis that you do, and the risks from SARS-CoV-2 are very high. So in general, you'll choose the vaccine if you're thinking about managing your risks appropriately. You would choose the vaccine over not vaccine.
yeah, I mean, I've
Unless
gained.
you dispose, right? Unless like you have certain pre-existing conditions that would say otherwise.
Yeah, I mean, I was about to make a joke about how I've gained weight since the vaccine, and I'm gonna blame the vaccine for that. But
Okay,
but
let's
yeah,
push on.
push on
Um,
to yeah. Now we're moving away from the coronavirus papers to more general bioengineering kind of papers.
mhm.
Uh, so
This one first, high throughput functional variant screens. Okay, so this one is I got the preprint for this. There's actually like I have like a mini story here. But, like I was looking at papers and I was like, oh, this is really cool. We try to cover open access papers, right? On our podcast, on our show, because those are the papers that people will likely have access to. And also, we wanna draw attention to the fact that opening up the publications of your works make it more accessible to everybody to talk about, and we'd like to see more of that in the world. So this one, it was published in PNAS, but I couldn't get the PNAS paper, but you can find the preprint. And that's like one of the cool elements of this whole preprint world is that it also helps us get access to literature. They may not be like the final version, right, that ultimately gets published, but you get to see earlier versions of it. And in this case, I'm not even sure. Sometimes people even update their preprints after publication to reflect the things that get published as a way of giving the scientific community, us, anybody who wants to read the paper, access. And so in this one, they're talking about, like, I didn't even know about this, Retrons. Have you heard of Retrons before?
no, I learned about them this morning when I was reading this paper. They're segments of bacterial DNA that undergo reverse transcription to produce fragments of single-stranded DNA, and it has antiviral functions, so sort of like the way CRISPR, but it kind of interferes with viruses.
Yeah.
Um,
And
uh,
so this mechanism there's some understanding this mechanism exists in bacteria, and so the authors want to use this technology, as we do in molecular biology and synthetic biology, to create an effect that we want in the bacteria. And the effect that they want to create is mutant libraries. Specifically, I think they want like SNPs. They want like a lot of single nucleotide changes. So in the past, one of the ways one would do this is dosing radiation or a mutagen of some sort, and that sort of creates it randomly anywhere in the genome. And the idea is that if you titrate down low enough, then the random event is guaranteed to be, or statistically likely to be one in a certain number of bacteria. But no one wants to work with those types of things anymore. Plus, that's like not super reliable because you can't actually guarantee the single mutation in one thing, and it's not easy to do those types of titrating experiments. And so in this one, they use these retrons to achieve the same effect and create a library of bacteria that all have a different mutation in it. And the reason why that's useful is because it lets you screen through those mutant libraries to try to find something of interest.
yeah, it allows you to like break different mutations, and they have this barcoding strategy as well, where they can sequence this entire library, so they can figure out where the mutation is. so that
Mhm.
if someone says, "Oh, this gene's interesting," we have it in our library. Do you want to look at see what the mutation does? That's a very powerful technique.
Yes, yeah, yeah. Knowing where, yeah, I guess that is an important thing to keep in mind because, like, yeah, you can make those mutations randomly, but then sometimes you have to, if you can't fetch them out again and then analyze them and follow up on them, then sometimes those there's a lot more work actually than that goes into working with those libraries. But yeah, barcoding strategy for a library gives you some very good insight into what's going on.
Yeah, so yeah, this is quite an interesting one. What? Oh, so next paper is something about, well, it's very much on ethics. So private information leakage from functional genomics data, quantification with calibration experiments, and reduction via data sanitization protocols.
Yeah, this is I like this paper a lot too. Also found it in the same way. It's going to get published in Cell, but this is the preprint version of it. And it actually talks about like we have all these big databases of gene information, sequencing information these days, and they're largely behind firewalls because you don't want nefarious actors to be getting in there and figuring out like how. Like what disease, what pre-existing conditions you have, essentially, right? That's like that would be a breach of privacy. So
Hmm.
that data is largely held behind um, uh, protection. But that also introduces difficulties in terms of who gets to work with that data, right? It sort of gives you like an institutional bottleneck where you have to be verified in certain ways to use it. It makes it difficult for the research community to use it. Largely, there's some data accessibility issues that come with holding data in that way. Oh, sorry. Oh,
Oh,
that was
so
just
no,
hitting.
sorry, sorry. Oh, no, I hit my microphone accidentally. But um, uh,
So the authors of this paper are sort of interested in better understanding what is that privacy risk, right? In terms of how could a nefarious actor link your information to one of these databases and then uh, through that means learn about one of your pre-existing conditions. And once you know how that attack can be done, how can you then change the data basically using maths, complex math, so that the data set can be released widely, but though that sort of attack on your privacy can't be accomplished. And so I thought that was just a fascinating concept, right? That you could essentially encode these type of genetic data so that the personal information can't be discovered, but you can still learn things about the disease, right, and the
yeah,
association with different disease variants in the
eh,
genome.
it's something that I often think about, like when in the like scientific publishing world, because we have to think about how much data we reveal about our patients as well,
Mm-hmm.
uh, and anonymizing data is getting harder and harder because in this age of digitization, it's becoming easier for people to like scrape data and then make correlations.
Yep.
So the threshold for what we need to do in order to make something anonymous is getting much more like higher, and
Yeah.
and genomics there is a lot of data there that can be extrapolated, especially if you're doing even if you're studying something completely different and you're taking all the genomic data, someone can pull out data on like various like diabetes or skin color or various other things that could potentially reveal who participated, which is again
Yeah.
quite a sensitive topic. And
Absolutely. And I mean, oh,
sorry, I was gonna say like
what?
the way we get around this at the moment is by getting researchers to sign contracts to make them liable, and so that if they do release information or something like that, they do get kind of punitive action. But
Mm-hmm.
again, that process of getting people to sign legal documents and go through this process takes time. And if you say in the middle of like a disease outbreak where this data it needs to get out very fast for people to analyze it and deal with it in time, that
Mm-hmm.
suddenly becomes a massive bottleneck, and that can
Yes.
be a problem.
Yes. And I also think it's not like, to me, that's also not the just way to deal with it. Like, also, research is done in the public sphere, right? There is
Mm.
an impetus to make things more public. It would be it's sort of, to me, it's a shame if the arguments of like that these privacy concerns aren't being, that these are real concerns, and but we shouldn't have to sort of block people from access, right, to research. Uh, uh, from these concerns if they
Mm.
can be circumvented and right if they can be addressed in other ways. And I sort of I think this is an interesting paper in that sense. I
Yeah.
heard that this particular technique is already being used by like I think the encode. Like there's a lot of big initiatives right going on in science to like do lots of sequencing and like to help be a research tool for other scientists. And they have already adopted this technique and have passed their data through those things so that they can make that data more accessible. It is public data, right?
Mm.
But not be vulnerable to the types of attacks that this author is describing.
Yeah, so that's really good. So this is quite an interesting paper to delve into then. Uh,
Hmm.
so moving on to the next paper, which is SSPABCD dash SSPFGH contributes a new type of DNA phosphorothioate based bacterial defense system. That's a mouthful of a
Yeah,
name.
I think this actually, to me, this also like groups with the Retrons paper, where it's like, uh, I think I would hope that people have a renewed interest in all the cool systems that exist in bacteria, because you never know when one of them might become a very interesting biotechnological tool. And in this case, these authors describe this type of bacterial defense system, essentially a way of targeting unknown pieces of DNA in the bacteria and targeting them for degradation.
Mm.
And it kind of interestingly, the way that it cuts these pieces of DNA is with this phosphorothioate,
Yeah.
phosphorothioate, which I think is like sulfur that gets attached to the backbone.
Yeah, yeah, it
Yeah.
attacks the backbone, which is interesting. It's in a new kind of way than what we've seen before. So let's see if I can pull out some figures for this. Uh, so
Yeah.
yeah,
I don't know if that's like easy for people to understand that small diagram, but that's how it is. Like, the DNA backbone is made of phosphorus, right, all joined up together. And this system, they find it adds the sulfur to the place that it's sent. Like, there's a sensing sort of object. It adds the sulfur there, and then that would cause the break.
yeah. So this seems to be yeah, and the interesting thing is that, like, can it have some function in bioengineering? This is
Yeah.
exactly the kind of thing that bioprospectors want to find, like something that does something new and interesting, and then think about what are the applications for it.
Absolutely. And but at the end of the day, or like sort of that's sort of like how people can use it. But and, for people who just love microbiology, I think this is also a very fascinating sort of thing because we know that phage and bacteria are constantly in this like arms race of figuring out like who's going to eat who, or like who's going to get infected and blow up your population and who's going to survive. And so like just learning about the complexity in this evolutionary arms race, I think is like very interesting.
Yeah, I mean, this is something for people who want to manipulate bacteria, because if you want to get a DNA into a bacteria, you have to jump over lots of different hurdles of all these different systems
Oh,
that
that
try to
too.
stop you.
Yeah, actually that's a really good point. This is also like learning about these systems is also really important in just engineering bacteria for research or industrial purposes, right? Where if you want to use a bacteria that isn't an off-the-shelf, well-studied thing, you're going to encounter a bunch of mechanisms that will limit your ability to work with them. And understanding some of these general things, this could be the answer, right? If you've been trying to transform your bacteria and it's not working, look no further. This may be this may be the reason.
Right. And next paper we've got is uh, highly parallelized droplet cultivation and prioritization of antibiotic producers from natural microbial communities.
Hmm. I super like this is almost on the coattails of hearing about that deep learning method to find antibiotics, right? You can say, oh, one way to get antibiotics is to make a whole bunch of chemicals and then figure out which one works. But we also have a history of finding organisms in the environment that make the chemicals that we want to work. And so they essentially use it's almost like this is the single cell sequencing technique, right? Of encapsulating things in little oil droplets, but they encapsulate cells, bacterial cells, into these little droplets, and then they're able to screen them whether or not they make any compounds that inhibit microbial growth.
Yeah, I'm quite excited for this one because the implications of microfluidics in kind of yeah, you're right about the whole way that we've discovered antibiotics, so throwing things against the wall. But this one, it evolves from just growing things in petri dishes to growing things in tiny micro droplets, and
Yeah.
so Yeah,
Yeah,
I
no, it's
mean,
the same. We're still throwing things against the wall. It's just like a different set of spaghetti, right? Like the deep learning method is like the spaghetti is chemical diversity, but here the spaghetti that we're throwing against the wall is biological diversity, right? Like going into like all the different organisms that grow. And we found them here before. Maybe there's one that we haven't grown yet that is gonna be the key. And yeah, I love that image. I love that image of just like the dots going through.
yeah, I've been watching these videos and I'm like hypnotized from them because it's just so satisfying to watch these droplets form, and each of these are different individual bacteria being separated into different things. It's
Yeah.
a lot of potential benefits for this. I mean, not just for finding antibiotics, but also other forms of bioprospecting, and also for things like diagnostics. If you have a diagnostic where it relies on growing a bacteria, that could be quite important. And
Hmm.
also for like testing out different like environments for bacteria to grow in as well. So
Yes.
rather than like
Yes.
having a massive like incubator, if you can have like a smaller incubator based on heat blocks to test out different temperatures where bacteria are comfortable at, that could also be really interesting. So
Yeah, absolutely. I mean, I remember like this in this vein of thought, like looking for unique antibiotics in environments. There was actually a technique that came before this. It's just a small tangent where like they had cells like sort of like etched holes into like a disc, right? And like you could have like tiny 96 well plates in small little things, and then you'd fill them up so that one bacteria would go into every hole. And so,
hmm,
like that's harder. I guess these days microfluidics is the way to go with that. Like you don't have to etch tiny holes and put bacteria in them. You actually just encapsulate bacteria into bubbles.
yeah. So again, this is very much an impressive techniques paper with a lot of impressive thoughts for if we take it into kind of other applications. So
Yes.
and also I love the way that eLife presents their papers. They're so easy to read. Uh,
That's the second
that's
that's the second time we've said nice things about
I
you. Like,
swear they're not paying me a single penny, but they are giving me papers to read for free, so I guess that's probably a bias.
absolutely.
So that brings us to PLOS pathogens with RNA thermosensors facilitate Streptococcus pneumoniae and Haemophilus influenzae immune evasion.
Yeah, I
Uh,
threw this on there because it's a recent pathogenesis paper. We're both
yeah,
pathogenesis people, and in this one they look at RNA thermosensors. So they actually find like just a tract of RNA that seems to respond. Like the gene, they know that this gene responds to temperature, and then they track it down to like the transcript that somehow part of this transcript is what's regulating its expression. And yeah, a story about
yeah.
thermal regulation.
Yeah, so the idea is that this RNA it has a specific shape, and at certain temperatures, it kind of opens up so that it can allow some DNA to be transcribed. So
Good.
yeah,
Allowed the protein to be made from the RNA.
and the interesting thing is the story here. The idea that bacteria prepare themselves for when you become feverish, and they have a specific like set of defenses in place, and it uses that it applies those defenses when it senses your temperature increasing.
Yes, yes, that's right. Yeah, I mean that's going back to this is the bread and butter of pathogenesis almost, right? Is that like we know we have defenses and we know that they're trying to get around our defenses. If we understand how those work, then maybe we can understand how to perturb little things, right? To make our outcomes the more favorable ones because I don't want to die from a bacterial infection. No, nobody wants to die from a bacterial infection.
No, definitely. And the last thing on the docket comes from actually a viewer. So we got a comment on one of our papers. So we do have like a library of articles that we look at, and in one of our previous discussions, we looked at a paper called evolution of antibody immunity to SARS-CoV-2, and
Hmm.
we barely skim these papers most of the time. So there's a lot of stuff we missed, and
Yeah.
this reader, Minimal, has actually brought up quite a lot of interesting things that have happened since this paper has been published. Uh, and
Absolutely. And also
yeah,
just putting in like nice clips from other scientists talking about similar sort of findings. And to me, what it sounded like Minimal is asking is like, how does this particular paper, Evolution of Antibody Immunity to SARS-CoV-2, speak to replicating reservoirs of virus in our bodies. And so like that's the idea that like after the infection gets cleared or like your productive immunity is up, you actually have places in your body that still have some virus holding on.
Yeah, so as a quick reminder, this paper, so the title is not necessarily the best title. Evolution of antibody immunity to SARS-CoV-2. It kind of implies a lot of things. It's not really specific, but I think this basically talks about how the antibody levels are maintained, and they link it to this idea that antigens are being maintained in the body, and that allows antibodies to change.
Yeah.
But there is this whole story that we've been nibbling around the sides of that there could be a long-term like SARS-CoV-2 infection happening in certain parts of the body that we've kind of missed, but I guess that touches on like what we talked about last week about exhausted B cells and why those B cells were increasing
Yeah.
after the initial infection has been resolved. So I feel
Yeah.
like
So yeah, I'm not sure. Again, it's like this is one of those things where this is to me this is how one reads papers when they're in a research field, right? If you're interested about a particular question, you read a bunch of different papers. They don't all have to specifically do with your question, but they have small pieces of data, right? That advance your line of thinking, and then eventually maybe someone writes a review, maybe you write a review, right, that brings it all together, or somebody actually finds from that initial inkling of what's going on, someone designs an explicit experiment, right, to address what all these little inklings have, and that's how you get a new paper. So again, like I think it would be interesting to choose this paper just because, I mean, we've heard a lot more now about evolution of antibody immunity to SARS-CoV-2, but it also might be interesting just to do like a short little segment maybe in our next news series where we go into maybe what do we know about reservoirs, right? Because we're always looking for different data out there. We might actually find some papers if we're searching that specifically speak to this.
Yeah, because I mean, we didn't choose this paper initially because it didn't really stand out to us, and I feel like I still don't necessarily want to choose this paper, but I feel that there is an interesting story here that we can tell that involves this paper. And so I think that
thank you to our one of our viewers for showing us an interesting potential place to search for literature, right? In terms of something that we could choose next, and we'll talk about it our next news week.
Yeah. I mean, the SARS-CoV-2 literature, there's so much there. I feel like there are plenty of stories that we are missing out on sometimes. And it's nice to let us know, if you're listeners, what you're interested in, so we can maybe focus in on that on occasion. I mean, we try not to be like kind of assassins on papers that we find to be terrible. We always try to find the nice parts of papers, but I mean, sometimes there is an interesting story to be told, and so I think we should set some.
I mean, I think at the end of the day, I've said this before. Like when you read papers, like you're always trying to know what you're learning from it, right? There's limitations to every experimental design, right? Like the science is not like they showed you and now, right? Like you're wrong and I'm right, and like let's move on. It's like, yeah, you're wrong and I'm right, but like, have you thought about it in this lens? Have you thought about it from this angle? Because the world is not like a linear story, right? And papers are stories. They're stories supported with data, but at the end of the day, you can tell a different story and you can pull in data from different places. That's why we do this channel, I think, is because only through reading the stories and looking at that data can you start to actually discuss scientifically, right? Like come up with ideas and like see the possibilities. Makes you also helps searching, right? Because like as you read in different topics, you then start reading in other topics, and it can send you in different paths to find out the information that you want to know.
Yeah, so I think we've agreed we're going to set some time to discuss this question in a bit more detail in 2 weeks' time as part of our news shows.
Yes.
But for next week, we do have to pick a paper.
Yeah. Yes. So we already did SARS-CoV-2 last week, right?
Right.
So I'm itching for something that is not that. I definitely I like that you've shown that video enough. It's almost hypnotized me into wanting to choose the
microfluidics paper.
Yeah.
Yeah.
The other thing that I'm also interested in. I'm also interested in the the Retrons one. Although I guess I wonder if
Hmm.
maybe one day that will be published in its full form and we can pick it up. Uh,
Yeah, I think so. I mean, can I tempt you with a malaria paper
the malaria
potentially?
paper.
Just because,
I am somewhat tempted by the malaria paper. I was looking through it a little bit more because I was interested because like new vaccine, right? I was like, oh,
hmm,
how does this vaccine work? But the paper doesn't tell us about how the vaccine works necessarily, right?
true.
It's a phase 2 trial.
Yeah.
Uh,
So yeah, you're right. We once you've read one phase two trial, you've kind of read them all. But
but it's an interesting technology. I almost wonder because we could do it, and we could maybe pick up the paper where they also did the technology. Like the technology, I find is very interesting.
yeah,
Use like some sort of viral antigen that makes nanoparticles, and they fuse that to their antigen of interest. So again, they're making nanoparticles of this antigen, but they do it through this viral protein. I thought that was kind of interesting. Um,
yeah. And there's also that modeling paper, which the time dependent uh,
oh yeah,
I can't, yeah,
heterogeneity.
had
Ah,
that. So
I am interested in it. It again, I'm always a bit a bit intimidated sometimes choosing things that have equations in them.
I feel like this would be too big for us to bite off. I think because it doesn't. It's not just like one paper. It does actually touch on quite a lot of other. Because I tried to dig into this, and even skimming it, it was quite hard. But
Yeah,
it's one of those papers I really want to understand, but I
yeah.
know it'll take me longer than maybe 2 weeks to get through it. So, uh,
I mean, we'll keep it on the docket, right? Like I
yeah,
always keep like a little bit in the potentials folder of stuff that like still seems interesting. I'll make sure I don't remove it, so it's always just kind of like haunting us if we ever want to do
yes,
something. If we see like another model paper, but we're like we want to talk about this model paper instead. I actually did remove like there were some model papers that were in here. One that was about like vaccine stuff I removed. So like yeah, this one can take the place there.
yeah,
Uh, and then I'm also interested in the phage one, the single-stranded new type of DNA based bacterial thing. Just because it's like I don't know, it's like a I like the story. It seems like a very straightforward story in terms of bacterial pathogen or not pathogenesis, like phage bacterial relationships. Um,
yeah, yeah. So that could actually be quite interesting as well. Um, so I mean, just looking at these from a perspective, from my perspective, you're right. The high throughput paper is this looks so good. It just there's there's so much like practical applications. I think for people who are working in microbiology and are thinking about especially bioengineering and
yeah,
bioprospecting, it just it
yeah,
there's a
it's like it's very like one of the reasons why maybe this speaks to microbiology nerds, right? But like
mm,
one of the reasons I got into microbiology was the fact that you can do like because they're single cells, right? You have full control of the genomes. You have this like screening capability, right? Like one of the things we always talk about bacteria is that like you plate them on a dish, right? Every colony arises from a single bacteria, and that was just like that's amazing to me. And seeing the way that they use microfluidics now to like basically extend that to now like don't worry about making a plate, you can make like like a continuous stream of plates even, right? Each one that has a unique thing from the environment. Yeah, that's really cool to me.
yeah. So yeah, I
Plus
would
videos, right? Like
yeah.
I mean it's hard to say, I mean it's hard to understate, like we are influenced by the fact that we're on YouTube. It's nice to show videos to people.
Yeah, and we do have to read and review these papers, and if they're set out in a way that is interesting to read, then I'm more likely to want to read it. So we are human. We are vulnerable to clickbait sometimes. So
But that's also more of a reason for our viewers to try to interact with us, right? Like you can sway us for sure.
I mean, they already have. In 2 weeks, we will be delving deep into a SARS-CoV-2 story. So uh,
Absolutely. Okay, let's do this. Let's do this immune or this microfluidics one. What's the title?
yes, so the title of the paper is I'm scrolling up quickly because I just kept was reading this through one. I just couldn't stop myself from reading it while we're doing this. So it's called Highly Parallelized Droplet Cultivation and Prioritization of Antibiotic Producers from Natural Microbial Communities. So I need to remember how to pronounce parallelized when we look at it next week, because I'm not good at pronouncing that.
Right. Yeah. So next week, join us for our deep dive week, and we will be covering that paper.
Yes, indeed. And yeah, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over on Twitter with the hashtag #MicroTWJC. We both believe that peer review is this process. I basically said this before, right? Like, we're all in on it. We want more people to join us. It's easier together. So if you have something to add, please let us know.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. See you then. Bye.